Yeah. So now we are going to have the last talk of this meeting by Professor Madan Rao, who will be presenting his last of the 3 lecture series.
Yeah, thanks. Yeah, I mean, the sort of thinness of the audience must have indicated that I've driven everyone away. And hopefully they will come. But in any case, um, so today, I mean, yesterday and the day before yesterday, I talked about uh, kinetic control of the size of the Golgi. And yesterday I talked about the mechanical control of the size of the Golgi. And those two ways of looking at Golgi organization gave rise to different insights, which one could not capture from the other. Okay? So it is important to first go through the kinetics and then through the mechanics. Uh, of course, doing mechanics is much more complicated because one has to include the mechanics of the fluid flow, and that makes the subject more difficult to study. But as I hope I convinced you, it gave rise to an insight which actually explains, for instance, the physical basis for cistern movement. Okay? And that's, though difficult, is a good thing to try and get. So yeah, so today I'll just speak, Sandhya, who has just left today, she was saying that my second talk was different from my first talk because I didn't give any gyan. So I'm going to give some gyan. I'm going to give the gyan which I forgot to discuss yesterday. And so I'm going to first start also with, I also want to, apart from giving gyan, I want to also talk about a few things that I feel I did not explain well. Yeah? So I'm going to just very quickly talk about that. So the first thing is the following here. Remember yesterday we talked about this Golgi membrane, which is being subject to fission and fusion. And the fission and fusion was incorporated in terms of active stresses that act on the membrane, and that those active stresses generate, in combination with elastic stresses, generate flows both on the membrane and in the fluid, and that in turn generates shape changes, including movement of the membrane. Okay, so that was the summary of the last talk. The biochemical cycle that you saw, which was associated with each active process, each active chemical cycle, involved a finite loop current, which went around the cycle. And as a result of the finite loop, the non-equilibrium loop, one was able to transport both membrane area and bulk volume, but also transfer momentum, which gave rise to these active force dipoles. Okay. So I just wanted to state here, address this following question. What is the equilibrium limit of this theory? Yeah? And there are two interesting equilibrium limits to note. One is, of course, to say that I restore detailed balance in these chemical cycles. When you restore detailed balance in the chemical cycles, that will make the loop current 0, and you won't get any of these fission and fusion processes. So that is one simple and obvious limit, the equilibrium limit. The other equilibrium limit is more subtle. This has to do with the statistics of vesicles coming in and going out from the mother membrane. Just imagine that the time scale tau, which we declare to be a millisecond, is very large, right? Let's say it's an hour. So every once in a while, every 1 hour, one vesicle comes and fuses. Then after an hour, another vesicle leaves. Okay? These time scales, given the time scale between fission and fusion events, is much larger than the shape and the density relaxation time. So between events, the system is relaxed back to equilibrium. Okay? So there's the other equilibrium limit of this. But this is a different equilibrium compared to the first one, right? This is an equilibrium where, as time progresses, you're changing the membrane. Okay? So in some sense, the area and the volume, okay, and other properties of the membrane change, okay, upon each event that happens. Okay? So it's a very long, slowly varying energy landscape that you're moving. Yeah? But that can be treated by equilibrium physics considerations. So these are the two limits that I wanted to talk about. Okay. The other question had to do, maybe when Neer comes back, because this had to do with a question that Neer asked. Okay. Now, this, as I showed you, is the phase diagram. Okay, is the phase diagram between the fission rate and the ratio of the fusion rate by fission rate. And what you see here is a stable phase when this parameter zeta is smaller than this critical line. Okay, and above that, you find that there is a spontaneous drift. The whole system now moves. Beyond this dashed line, you get an L equals 2 instability, namely it becomes an oblate ellipsoid. It's unstable towards that chapati-like formation. L equals 3 instabilities, L equals 4, and so on and so forth. So in this region, okay, in this region, which includes the green one and this pink piece here, okay, you have spherical shapes which are stable. Okay, so in the green one, you have spherical shapes that are stable. In the pink one, in this region here, you have spherical shapes that are moving. Yeah, that's the other prediction that comes out from this. Okay. Beyond that, they are no longer spherical shapes; they are unstable to shape deformations. Okay. That is that statement. I also wanted to say the following: that the result of this is saying that at very high L, that corresponds to having lots and lots of tubules, okay, which is only limited by the bending elasticity. Yeah. So at these large L deformations, the system, the dynamicals, the system here has many prongs or tubules coming out. Now you can ask whether that configuration can also be achieved by purely equilibrium means. Can you have specialized proteins in the case of the endoplasmic reticulum? They're called reticulons. Okay, can you have specialized proteins which locally have a spontaneous curvature and generate these tubules? And indeed you can. Okay, one is not saying that you can't do that. You can't generate these tubules by equilibrium means. Okay? And indeed, these are included in our formalism because they reside in the values of the bending modulus and spontaneous curvature and so on and so forth. Okay? So that's the other statement. Okay. Yeah. Now I wanted to make this statement. Remember, in the initial part of yesterday, we talked about a nearly planar membrane. And we had vesicles which came in and went out, both a nearly planar version and a closed membrane version. And they gave rise to the following predictions, which also came out from the more sophisticated theory, but which are borne out by experiments conducted by various groups. Okay? And the predictions were that it forms tubulations and folds. You get negative tension instabilities. There's an effective dynamic tension, and there is a flattening of the closed membrane, and there's a drift. Okay? These were predictions that came out from these simple considerations that I talked about, without including hydrodynamics. Yeah? So why is it that we needed to go into hydrodynamics and do this very difficult theory? Right? More difficult compared to these simple attempts. And I just wanted to stress this point, that just because a theory agrees with experiment doesn't mean it's right. Yeah? And this is an important thing I wanted to tell you. Please, there's a article written by Feynman. It's called Cargo Cult Science. I think you should read it. And what is important here is that we could have been satisfied with this kind of description, okay? But there was a conceptual issue at stake, and we needed, namely, the violation of momentum conservation, and we needed to address that issue. Okay, and so it took us that much time to and that sophisticated analysis which Alex Rautu had done in order to address that issue. Okay, and I believe this is a very important point that I would like at least the younger students to appreciate. You should bend over backwards to try to show your theory can be violated, can be wrong. Okay, and yeah. Yeah, sorry. Yeah. Ah. It could be, of course. Yeah. No, yeah. Exactly. No, I was just, I guess, responding to something that Neer and I were talking about during lunchtime. And it's true that people like Misha Kozlov, whose theory this thing is, talks about how the tubulation in ERs are generated by these specialized curvature generating molecules called reticulons. So this is Rappaport and Kozlov. Right? And all I'm saying is that the phenotype of having lots of tubulations is not restricted to a non-equilibrium phenomena. You can also get it in equilibrium. But the non-equilibrium phenomena, as I said, that we are describing here is thrust upon us by the fact that vesicles are coming in and going out at a very brisk rate. Right? And so you have to, whatever you do, even if you have reticulons or don't have reticulons, you need to address this issue. Okay, and then since Neer is not here, I'm going to the second part here. Remember, Neer had asked how is the x-axis, the fusion rate divided by the fission rate, and if the value is greater than 1, you would have more fusion than fission, and that would increase the mass of the vesicle. Yeah. And so the precise statement of this is that this is just the rate. Okay? This is just the fusion rate and the fission rate. This doesn't tell you what is the mass transfer. The actual mass transfer is the fusion rate multiplied by the area fraction of the fusion proteins. Okay? In some places they could be available, in some cases they are not. Yeah? Minus the fission rate multiplied by the area fraction of the fissioning proteins. Okay? So it's this, in our analysis, we hold this to be fixed throughout the calculation. Yeah? But that doesn't mean that the fusion rate by fission rate could be they could be greater than 1 or less than 1. Okay? Yeah. Okay. Then we have something here. Yeah. So now, so this one says that there is if you have high enough activity parameterized by zeta, then you generically get the spontaneous drift. And so when you look at mammalian cells, you apparently don't see any spontaneous drift of the Golgi. Okay? So what is the reason for that? There are two possible reasons that come out from, two possibilities that, that appear from our analysis. One is that it's possible that if zeta is smaller than this critical value, you have a stable configuration. Okay? So maybe the values of zeta in this context lie in this green region. Okay? Or a more interesting possibility is that there are other factors that come into play which actually fix the Golgi cisternae. Okay? And that could be the cytoskeleton, which we have ignored in this calculation. Okay? There's an actin cytoskeleton and a microtubule cytoskeleton that can provide scaffolding to these organelles and hold it in place. And indeed, we know that there are proteins that bind from the Golgi to microtubules or the actin. Okay? And so that's a possibility. And now you could say that, oh, if there are indeed this actin, the cytoskeleton meshwork surrounding and trapping this, then it's possible that hydrodynamics is unimportant, that you did the calculation in vain. Yeah. Of course, it's true that if I put an sort of actin mesh around it, it will screen hydrodynamic flows. Okay? And that's certainly true. And so hydrodynamic flows will sort of decay at an exponential scale. How fast that is depends on the properties of the meshwork. But despite that, there is a suppressed instability which is waiting for release. Okay? There's an instability both in terms of shape changes, giving rise to this thing, or movement. Okay? And the moment you sort of disturb or loosen the scaffolding, possibly this will be released. Okay? And so you'll miss all that physics if you don't include hydrodynamics. Okay? The other feature I want to emphasize is that one of the consequences of our analysis is that we showed that the vesicle transport and the cisternal progression model are actually part of the same underlying physics. They are just different phases. So again, you won't be able to appreciate that if we did not include hydrodynamics. Okay? So I believe there is some virtue of going through a rigorous analysis, despite the fact that on the surface it might seem that one doesn't need to include hydrodynamics. Okay? But the fact is that it is there, and we need to. Okay, so I think I've finished my gyans for the day, and I'll talk now about what I'm going to talk about in this lecture. And so what I'll be talking about is primarily coming from this paper. Alkesh Yadav, who did all the work in this paper, is a student of mine who is graduating soon and actually going to Israel for a postdoc. And this problem has to do with a sort of information theory problem. Okay, an information theory justification for being able to sort of control the number of cisterna. So far, I've been talking about the size of the cisterna, the shape of the cisterna, etc., etc. But now I'm going to address the question of number. Okay, and what this calculation is about is how the number of Golgi cisterna, or the number of Golgi compartments, and enzyme promiscuity, okay, facilitates the synthesis of a very complex glycan code. Okay, and that's what this work is. This work will involve two interesting features. One is that it will involve a high-dimensional optimization, and the second that it will involve information processing and systems biology. I also wanted to flash another paper which has just been accepted in eLife. This is again by a student who will be finishing next year, Krishnan Iyer. And this is about using a similar kind of philosophy to address the question of how cells in a developing tissue infer their spatial position. Okay? So these are different kinds of questions that one can ask in biology, different kinds of theoretical questions that one can ask. And this is about what I mean by in the first lecture when I talked about how new questions arise when physicists look at the phenomena of life. Okay? These are new kinds of questions that one may ask of, for instance, these cellular and tissue processes. Both these ideas involve this high-dimensional optimization and an interplay between information processing and systems biology. So there's much in common with them. Okay, these two ideas sort of give rise to this notion of cell as a distributed system. This is something I talked about in the first lecture, right? And I just wanted to put this up here so that you appreciate what a distributed system is. I defined it in the first lecture, but here is a distributed system is a system of autonomous components. They could be anything. They could be cells in a tissue. They could be organelles within the cell, etc., etc. Okay, which interact with one another in order to achieve a global task. In the context of the cell, it's a cellular task. In the context of a tissue, it's a tissue-level task. Okay, but individual cellular components are taking independent decisions, collaborating with each other, interacting with each other in order to achieve a global task. Okay, and this is an interesting way to look at many phenomena in biology, especially in the field of control. Okay? How do biological systems control a variety of phenotypic features? Okay, so in this particular talk, as I said, I'm flashing this picture again. I'll be addressing the question of what sets the number of organelles, okay, or subcellular structures, and in particular, I'm going to ask what sets the number of Golgi. I'm going to propose a possible strategy to understand this. Okay, so again, the question, what sets the number of Golgi cisterna or compartments? And here is some statistics that you might find interesting. The number of cisterna in human cells is around 4. Okay, the number of cisterna in plant cells shown here is around 20. Okay, you can see that many more stacks of Golgi here compared to the cisterna that you can see here in that schematic, okay, which is of an animal, of a mammalian cell. in green algae, it turns out, even though they're pretty small, okay, that's the size range of the green algae cells. There, the claim is that there are around 100 Golgi cisternae. Okay, so that's remarkable. This diversity of number of these organelles is quite remarkable in different species, and of course calls for an explanation. Okay, so before we step into the functional arguments that I'm going to present, let me just put out here one proposal that was made by Svetina's group, which is purely based on membrane mechanics. Okay, and the claim is the following. We saw many pictures which said that the Golgi cisternae had these chapati-like structures, right? These flat pancakes, as they call it, right? And so let's approximate those shapes by these discs, okay, with a certain, where the larger diameter of the disc is d, and the smaller, the rim has a certain radius r of t. And let's assume that there are n of them. Okay? N could be 4 in humans, 100 in algae. Okay. And now I give you the following numbers. I tell you that this membrane curvature modulus, okay, is 10 to the minus 19 joules, okay, 30 kT, yeah, and the addition strength, the sort of sticking strength between this cisterna and that cisterna is given by 10 to the minus 16 joules per micron square, okay, which is around that number. Okay, so the physics is the following. There is an interplay between membrane bending, okay, and membrane adhesion. Okay, the bending for this has to do with like a disk, so you can use Helfrich theory to calculate that, right? The disk, I wish I had a disk here. Oh, I can draw it. Okay, and that's rotated circularly. Yeah, so this is flat. Okay, zero radius of curvature, zero radius of curvature. The radius of curvature of this side, there are two radius of curvature. One is this radius, which is d by 2. The other is this radius, which is r of t. Right? So I can calculate what is the energy of this configuration. The curvature, just look at the curvature energy alone. Okay? And I've given you what the bending modulus is. So I can calculate this exactly. I can do this for n cisterna, right? Just n times the curvature energy of a single guy. And then I had to calculate what the addition energy is. The addition energy is just this number. So minus, because it's attractive, minus this number multiplied by the area of that flat patch there, which is just pi d by 2 square. Right? So you know the full energy of this configuration, if these are the elements that go for the energy, membrane bending and adhesion. Okay, now you have to keep the total area of the membrane and the total volume enclosed in this in these things, in these stacks, to be a constant. So those are the constraints. Yeah. So I take the energy, take the constraints, minimize with respect to n. Yeah, and I should get some number, some finite number. That was the proposal of this paper, and the claim was that the number of cisternae is dictated by that energy minimizing strategy or energy minimizing approach. Okay, difficult to prove whether this is correct or not, and I'll leave it to you. What I'd like you to do is, because you know all the ingredients of the physics, you can do the calculation. So will you do it for homework? Because you don't need to show it to me. But if you can do the calculation, that just gives you some some confidence about your understanding. Yeah? You can carry out the full calculation and draw a phase diagram. Yeah? Okay. Yeah. So my criticism of this, I won't belabor this point too much because I don't want to address this issue at all. But I wanted to just state it that you should think of this also. The kappa C is roughly the same for lipids. It's around 30 kT for everything. And again, the addition and strengths are not too different from each other. So using such a thing, you need to be able to explain this wide diversity that you see here. Yeah? And I find it difficult to imagine how to describe this wide diversity. Okay? Yeah. 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 Uh-huh. Right. 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 What should be right? This is the first day's the model for the first day. Did it make any predictions for the number? Correct? Yeah, that's indeed that's exactly correct. Himani's the work I talked about, the aggregation fragmentation model. There, the dynamics of aggregation and fragmentation is tightly coupled to the chemical processing. So the number of cisterna that you get is tied to the number of A, B, and C species. Okay, so if one calls them different chemical species or different enzyme species, the number of cisterna there in the vesicle transport scenario just scales with the number of A, B, and C species. In the other scenario where I allow the cisterna to move, so the cisternal progression scenario, there the number of cisterna scale not only with A, B, and C. Actually, it doesn't scale very weakly with A, B, and C, but it mainly scales with the size of the system. And it goes as square root of the size of the system. That is the result of that calculation. But I think the calculation needs to be more sophisticated in order to address this question. But maybe one can, I guess you're suggesting that you can make the calculation more elaborate to try to address this question. You're right, yeah. Yeah. Okay. So what I'm going to talk about is functional demands that drive cellular complexity. This is going to be our take on the following, and we'll see how it goes. Okay. So what is the primary function of the Golgi? The primary function of the Golgi cisterna is in this process called glycosylation. Okay. What is glycosylation? Glycosylation is a sequential chemical processing, okay, that is attachment of sugar groups, okay, on top of proteins, and this happens in the cisterna. As a result of adding sugar groups sequentially on proteins, a picture of these sugar groups are like that. Can you see here? Okay. Yeah. As a result of that, you display them on the cell surface, and you see a whole display of what's called a relative forest of these branch-like structures called carbohydrates or glycans. Okay. So these are glycans. This process, this chemical process, the sequential chemical process that happens in the first cisterna, then gets transferred to the next, then it gets transferred to the next. The sequential chemical processing is called glycosylation. Yeah? Is that clear? So each of these cisterna are actually little chemical assembly lines, yeah, that have resident enzymes, which will then catalyze chemical reactions that involve a protein that comes out from ER and some sugar groups. Okay? They keep adding. Okay, it's like an assembly line. Okay. And then after some amount of chemical processing is done, it gets transferred to the next cisterna. More chemical processing happens, gets transferred to the next, and so on and so forth. Yeah? So that's the process. It's an incredibly complicated process. Yeah? I'd like you to appreciate that. Most of this process here, the creation of these very complex structures that you finally see on the plasma membrane are not coming from a template. They're not templated. It's not like DNA or proteins, right, which are templated objects. Correct? They require a DNA, there's a DNA background, there's a RNA code, etc., etc. Right? There's a template on which that acts. So I can do, for instance, PCR, and take something from one cell and just enhance the production of that, and I can do experiments easily. This is not templated. This is happening by stochastic chemical reactions. Okay? And yet what you find here is a very precise display of molecules, which are markers of that particular cell type. Two different cell types, namely neuronal cells or skin cells, have a totally different kind of display of glycans here. Okay, that's interesting, isn't it? So, that's what happens. Okay, and that's the primary goal of this Golgi. The primary goal of the Golgi, to repeat, is being regions, spatial regions, which house different enzymes, which are places where this chemical assembly line goes on. Okay, so now let's see what does that. Let's now go step by step and try to see what happens. Okay. Now, what is the use of these glycans? The glycans are paradoxically involved in many, many different kinds of functions, multiple functions. Okay, they're much more. I mean, despite their diversity, they're hugely diverse. Okay, but despite their diversity, there's been, unfortunately, not been as much attention has been given to them as to proteins and nucleic acids and so on. Okay, the diversity of glycans and the diverse functions that they play should actually, should sort of provoke more interest. And I do hope that many of you will sort of think about it. One of the daunting things, of course, is the very complex chemistry. And a lot of us, while we might have left, biology in the 10th standard, we must have left chemistry maybe earlier also. So, but I think it's useful to think about chemistry, chemical processing in terms of information processing, a different way of looking at it, and it might give you more interest in the physics there. Okay, so what you see here, is this dense forest of glycans that I'm seeing outside. Okay, in blue, this is the cell, and this is the outside of the cell, and you see the dense cloud there, made up of many, many, many branches, hairy structures, flexible polymers, flexible branched polymers that reside on the cell surface. Okay, and so here are some of the features. All cells in nature have a dense outer coat of glycans. Okay, all cells. Glycans dominate the interface between us, namely the host, and them, namely pathogens or bacteria. Okay? Glycans play a key role in human, pathogen, and cell-cell communication and regulation. The large complexity that is encoded in glycan structure is non-template driven. Okay? That's what it comes from stochastic chemical reactions. Every disease that affects humans significantly involves altered glycans. So this is more than enough reason to study them. There's really complex things. They're involved in all kinds of different cellular and tissue and organismal level functions, such as cell-cell communication, cell pathogen communication, cell pathogen evasion, cell niche recognition, etc., etc., etc. Okay. Finally, it's also involved in cell identity. Again, a feature that I'll come to in the next. And so you can see this is a sort of a schematic of the many varieties, many sort of diversity of functions that involve these interesting molecules called glycans. Okay? So you should think of a given cell which has a cell surface, which is decorated by these diverse glycan profiles. They display to the outside world a molecular information coding. Okay? And so there is some pattern recognition that needs to happen from one cell to the other, from a pathogen to the cell, and so on and so forth. So I'm using these words intentionally so that I sort of trigger some excitement in you to bring in new ideas coming from information theory. Okay. Now, in order for the glycan distribution to actually realize those enormous number of functions, the glycan distribution needs to be both diverse and complex. Okay, and diversity, to distinguish these two, the diversity has to be just the number of different kinds of glycans you can produce, the chemical diversity. The glycan complexity means that the distribution has to be really, really like so. Okay, and I'll try to make that precise in the next talk. Okay. So you can see sort of anticipate this, because the glycans have to be, that is produced by a single cell, needs to be strongly anticorrelated for pathogen invasion, anticorrelated to the receptors and to the molecules that are sitting on the pathogen. They need to be also strongly correlated with the correct niche. There's a skin cell should be in the skin, the neuronal cell should be in the brain, etc. Those are different niches. Okay, you can't have the neuronal cell in the skin because then you sort of trigger an immune response. Okay, and that's being done by glycans and so on and so forth. So it needs to be strongly correlated with the correct niche and anti-correlated with the wrong one. Okay, such functional complexity drives glycan complexity. This is the proposal and the approach that we are taking. Yeah. This is a famous article written by Ajit Varki, one of the leading figures in glycobiology. And this is called the Red Queen effect, a phrase that has been derived, has been taken from Through the Looking Glass, Alice in Wonderland. For those of you who haven't read this remarkable book, please read it. It's very, very, it's extremely interesting. And one of the phrases here says, so that's the Red Queen taking Alice. Now here, you see, it takes all the running you can do just to keep in the same place. If you want to get somewhere else, you must run at least twice as fast. Okay, and what does that have to do with this with glycans? And here is the thing: imagine that you have host glycans. The host glycans mediate intrinsic functions, a variety of functions, including pathogen binding. Okay, binds to the pathogens. The pathogens evolve to bind to the host glycans because they want to try to enter the cell. Okay, so they want to bind to some protein. The host with now, but the host wants to escape that, right? So it will try to change its glycan profile. But the pathogen evolves much faster, okay? So it'll change it to accommodate that new changed environment. And so you see, that's the Red Queen effect. The host is obliged to keep running in order to survive. Okay? And this is true for all the other functions that I talked about. Okay? So this is an interesting game theoretic, right? Game theoretic idea that has entered the lexicon of biology. Okay, so because glycans, functional complexity demands that you need glycan complexity. Glycans are produced in the cisterna, in the Golgi cisterna. So our approach is going to be that maybe the driving force for cisternal number is glycan complexity. That is, if I have a more complex cellular machinery, i.e. more number of cisterna, then that will give rise to a more complex glycan profile. Okay, so that's the root. Okay, so here is how do we define complexity, and this is something I'm just very quickly going to. These are two data. This is mass spec data, mass spectrometry data of the glycans from planaria. Yeah, these are small, tiny two-dimensional organisms, worms. They're called flatworms. Okay, planaria has around 50 different cell types. Okay, and this is the typical mass spec data from planaria. You can see that is not very. You would agree that this is not very complex. Okay, a few number of peaks. Yeah, and so in the complexity graph here, it has low complexity. Compare this to the humans. This is the human neutrophil data, and you see many more peaks. Okay, and if I sort of draw some kind of curve, it will go all the way around. Right? Some coarse-grained representation of this, I'll get lots of peaks and troughs. Okay, so humans have around 200 cell types. Okay, many more cell types than that. Okay, and therefore, if the glycans were markers of cell type identity, then I should be able to distinguish the glycan profile from these 200 different cell types. So it can't be a smooth Gaussian, right? Two Gaussians are very difficult to distinguish. Okay? Yeah. So the fact that you want to have a distinguishability in a complex organism sort of drives the complexity of the glycan profile. Okay? And the way to the quantitative way to define complexity is by doing a coarse graining of this data and using something called a Gaussian mixture model, which is just fitting these models with a finite set of Gaussians. Yeah. 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 Right. Good question. Yeah. Good question. Right. Right. So remember, I was saying that that the number of functions that need to be carried out, okay, are huge in all these things. Plants, for instance, use glycans to for large scale. Plants can't move around; they are sessile objects. So tissue integrity, tissue rigidity, etc., is a crucial part of their survival, and a lot of the glycans are employed to do that. So there are a lot more functions that are ascribed to glycans in these in animal. I don't know about algae too much. I'm sorry, but yeah, I know it looks like a simpler organism, but the number of functions that are carried out by their glycans could be enormous. Yeah. So that's I think what is happening. But that's a good question, actually. One of the things that we'd like to do is to actually follow, this set of arguments followed with real data, do some kind of bioinformatics data and data analysis. So this is something that we're thinking about. But that would make it at least slightly more precise then. Yeah. Yeah. If you're comparing sizes between organisms, is it? No, but between organisms is very difficult to compare. I mean, you can compare it, of course, but the cell size is very different. The amount of material inside is very different. So. Yeah. So right. So you can do such things if. Remember, I talked about limiting pool theory in the first class. So there, of course, is right. If you have two cisterna, for instance, in a cell with a limiting pool of material to constitute them, then if I have 2 versus 3, that will affect the size of this thing, right? But in the case of the Golgi, I don't think it's limiting pool that is responsible for the final. It has to do. The ensemble is where you have a constant flux of material coming out from the. So it's a constant rate rather than constant number. Yeah. Yeah. Different types of cells within an organism. Yeah. Correct. Yeah. Yeah. So, okay, I don't think so. I don't think the number of Golgi cisterna vary, but in RBCs are very specialized cells, so I don't know. After all, RBCs are anucleated; they don't have a nucleus. Right. Exactly. No, I know they have glycans, right? So they have to have Golgi, but yeah. So I would believe that within an organism they have the same. Yeah. Yeah. Within you could right. I understand. Yeah. So they are discrete guys because this is mass by charge ratio, right? So they'll be discrete. Exactly. Exactly. Absolutely. Right. So you could have complexity coming from exactly. Or the basic units could, yeah, exactly. So there are a variety of ways in which molecular complexity can increase. Yes, exactly. And so I just wanted to say that this is not the bare experimental data. This is denoised and thresholded, right? And because there's noise coming from the mass spec data, and there is some other, So when the experimentalist gives you this they will give a processed data. Yeah, correct. You could do more. Yeah, exactly. There could be there could be branching complexity. There could be, because these are branched polymers. The monomers could be different. You know. Yeah, there are many ways in which they can be. The complexity can change. Okay. Right. So yeah. So now the definition of complexity is going to be the following: what is the number of components of the Gaussian matrix, Gaussian mixture model that is required to approximate the target profile? The target profile is the glycan profile that is obtained from the mass spec data. Okay. This is the we'll declare that this is the profile that this given cell, in this case neutrophil, wants to achieve. Okay? And it achieves it by this stochastic chemical processing. Yeah? So the number of components of the Gaussian mixture required to approximate the target profile is a measure of the complexity of the glycan distribution. Okay? If it is a simple one, I just need, let's say, 3 Gaussians to approximate this to a given accuracy. With the same accuracy, I require maybe 20 Gaussians. Okay. And I just wanted to now distinguish between diversity and complexity of the target glycan distribution. So here, what I've drawn here is a schematic of a few coarse-grained glycan distributions. Going from here to here, I would say you've increased the complexity, you've increased the number of peaks and valleys, which means that you've increased the number of components of the Gaussian mixture model. Going from here to here, I've increased the diversity because this width in this direction has now increased. Okay? I have more number of, the glycan index is more spread. Okay? So that's diversity, this is complexity. Right? Okay. Okay, so now given all this preliminary thing, let me state the molecular encoding problem. We want to state the molecular encoding problem as an information processing problem. Okay? And here's the objective. So suppose I'm given a desired output, namely the target glycan profile that the cell wants to achieve. Yeah? That's what I want to achieve. So given a desired output distribution, P of G, G standing for glycans, of a given complexity, okay, what are the requirements on the cellular synthesis and processing steps that achieve the target glycan distribution with a high fidelity, okay, or high accuracy? Yeah? So this is the question I want to pose. Now you can see that since both the output target, namely that was obtained from the mass spec data, and the channel, namely the cellular synthesis and processing machinery, okay, both are noisy, they are subject to stochasticity, we have to use the tools of statistics and probability distributions. Okay? So that is going to be the language that we're going to use. Okay. Now when do we say that two probability distributions, here the target distribution and the empirical or synthesized distribution, are close to each other? Okay? So drawn here in the dashed red line is the synthesized distribution by the cell. Drawn here in purple is the target distribution that the cell wants to achieve. Right? The complexity is defined by the number of ups and downs that it undergoes. So you would say intuitively that these two distributions are far from each other. Right? And similarly here, you would say that these two distributions are very close to each other. Okay? So can you quantify this? Right? What is closeness or farness? Okay? To quantify how close the empirical or synthesized distribution Q, right, Q is from the desired target distribution P, you define the Kullback-Leibler distance. Okay? This is one metric or one distance measure that tells you how far is one distribution from the other. There are many that you can. This is a metric that is inspired from information theory. Okay? And the definition of the KL divergence, Kullback-Leibler divergence, or Kullback-Leibler distance between distribution Q and P is the following. Okay? P of x log P of x divided by Q of x. Yeah? So this is the distance measure between these two. How do we understand that? Right? This is a formula, but let's try to understand it. To get an intuitive feeling, let's plot the probability distributions. Let's take an example. Okay? This is P, having two humps here, and this is Q. P, remember, is the target distribution that you want to achieve Q is the synthesized distribution. Okay. Now we'll compute the terms in the definition of given here. Okay. Let's just rewrite dKl in terms of this quantity. Okay. I'm just doing simple algebra. So I'm going to plot minus log Qx and minus P of x. Okay. So I take this, minus log Qx looks like that. And for this P of x, minus log P of x looks like this. Okay. So what the dKl is actually the shaded region, okay, multiplied by the probability distribution of P, okay, integrated through all values of x. Okay. And that a small dKl will be a small shaded region, so they're close to each other. A large dKl will be a large. Okay. So that's how you define closeness and farness for a distribution. Okay. There is another way to interpret this dKl, and that's the following. You can rewrite dKl again by that formula that I just flashed 2 slides back. Rewrite it this way. Okay. And now this one can be interpreted as a difference of 2 entropies. One is a cross entropy between P of x and Q of x, and the other is the intrinsic entropy of P of x itself, the target entropy. Okay? So rewriting, this is just rewriting, trivial algebra. But what this allows you to interpret is the following, that the KL divergence, the Kullback-Leibler divergence, is the average log likelihood of samples generated from P of x to come from Q of x, giving a measure of distinguishability. Okay? So this is the, you can reinterpret this as an average log likelihood of samples. It's more convenient to define a fidelity, okay, which is the ratio of dKl by this entropy. Okay, because you're going to be using different target distributions which have different widths. So it's best to normalize it with respect to the target distribution. So this is indeed the quantity that we're going to look at. We'll just define this to be fidelity. Okay? So I'll stop the discussion for now, but please remember this because it will come later. Okay? Okay. So now that we have discovered what is a good way of saying one distribution is close to the other. But now I have to do the following exercise, namely, I have to tell you what is the cellular synthesis and processing model that I'm using. Right? I have to tell you what this is called channel. What are the physical and chemical characteristics of this channel? This noisy channel. Okay? And this is it. You have the ER, it produces processed proteins. They are transferred to the first cisterna, whereupon chemical reactions happen sequentially, PC1, PC2, PC3, etc., etc. Okay? Then they are transferred with rate mu to the next cisterna. Again, chemical reactions happen, and so on and so forth. Finally, after the nth cisterna, okay, nth cisterna, you deliver the goods onto the plasma membrane, and that's your final display of the glycans. Okay? So what does the channel model involve? The channel model involves one is the number of kinds of different kinds of glycan binding proteins that are synthesized and processed by the ER. This is what we talked about. It involves that. So it turns out that around 1/3 of the proteins that are made in the cell are sort of synthesized in the ER. Okay? And around 70% of those proteins are trafficked through the Golgi. So it's quite a high number of different kinds of proteins that are trafficked. Okay? And so they will all be these different glycan binding proteins. Complicated, obviously complicated. Okay? The processing steps involve the following features. I will declare the number of cisterna. In this particular case, is NC cisterna. I will tell you what is the number of enzymes that the cell has. Here there are NE number of enzymes. Okay. Then I will say remember enzymes have a certain specificity for a given substrate, right? Enzyme substrate interactions. A given enzyme has a given substrate, or a given substrate is catalyzed by a given enzyme. But often there could be enzymes which can catalyze 2 different substrates. Okay? So this is called specificity. Yeah? And so one has to declare what the specificity of enzyme substrate reactions are. Okay? One should also be able to partition, because the different cisterna carry different enzymes. So you need to partition the enzymes in different ways. Yeah? And the many ways of partitioning NE enzymes in NC cisterna, the combinatorial problem, will be one of the elements of the optimization. Once you have partition enzymes, you have substrates, you've declared the specificity of enzyme-substrate reactions, you have to talk about what are the chemical reactions that happen. So I provide to you a chemical reaction network with given chemical rates. Yeah? Then once the chemistry has happened here, they are transferred from one cisterna to the other with a rate mu. So I should give you the intercisternal transport rates. And finally, I have to fix what is the topology of the chemical network. Okay? So these are the things, are the features that define this channel. Is that clear? Okay. NS is the number of reactions that are happening in each cisterna. So, right. So, for convenience, it's just good to take that. Whether this reaction actually happens depends on the reaction efficiency of that particular, whether that enzyme is there or not. So, for instance, if some reaction does not, if this enzyme does not here, then this won't happen. So that can be, you're just giving it, you could have called this some infinity or something. You're putting a large number there. No, there is. So because remember the enzymes are different in different systems. Correct. But they have the same number of NS. So NS is the number of possible chemical reactions that happen. The actual number of chemical reactions that happen here depend on the number of on the number and kind of enzymes. Right? If you don't have that enzyme in this reaction, then that won't happen. So it will stop at some earlier value. But it's the same in all the systems. Huh? It's the same in all the systems? No. That's what I'm saying. So suppose I just have enzyme A and B here. Okay? And here I have A, B, and C. Right? Then here I'll allow for all three reactions to happen, but I'll only proceed up to A and B, and after that I can't proceed here. Whereas here I can proceed from A, B, and C, because I have all three enzymes. Piece of glycan. Yeah. So it's like building a polymer. Exactly. So except it's a branch polymer. Yeah. Okay. Yeah. But then it means that B can add more and more and more B. Right. B monomers. Yeah. Not just one B monomer. It's not really that B is distinguished as a single B. Yeah. Exactly. Depends how long you lingered in this system. Exactly. Which is controlled by the rates. Okay. Right. Exactly. Yeah. So already there, even with just one enzyme B. Absolutely. You can have a runaway polymerization. Yeah. Exactly. So that complexity with just one enzyme. Exactly. So you won't get any complexity at all. No, you can get. No, you'll only get runaway reactions. So live at different times. One left. I'll show you. Huh? Sorry. One substrate can live with just one monomer. No. So I'm assuming that the mu's don't depend on the index. The mu's are controlled by what happens in the membrane. So it's constantly leaving with a certain rate. Right. So all the mu. Yeah. So you could take away a protein, a processed molecule, before it has reached its desired thing, or waited for a long time. Yeah. So already you have a distribution. Yeah. But you won't get the complexity. I'll show you all these things. They'll come out from. Call that complexity? No. Let me. I defined what complexity is earlier. Yeah. Sorry. Yeah. No. So P labels the protein that is coming from there. So C1 is just the addition of a sugar. Then C2. PC2 is the addition of the next entity. Okay, so I'll come to all these things. They interact? No. They don't interact. Yeah. But that's not crucial here. That's not important. Whether they interact or not is not the important issue here, because that will be taken. Remember, these are chemical kinetic equations, right? So So they'll be, so if they're interacting, it'll appear in the rates. Right. So, I'll define all these parameters when I write down equations. But mu is the transport rate, and there's a set of many rates that control the chemistry. Okay. Yeah. So, okay. So the elements of this cellular synthesis machinery is the following. I have to declare the number of cisterna, NC, which is typically 4 in mammalian cells. Total number of enzymes, NE, typically 15 in each cisterna. Number of reactions in a cisterna, they're typically 10 to the 4. Okay, so you can see that an S, the number of reactions that happen in the cisterna are much larger than the number of enzymes. And there's a precise partitioning of any enzymes in NC compartments. Okay, so now here's the set of equations. Okay, so typically each chemical reaction is a Michaelis-Menten reaction. So you're having a sequence of Michaelis-Menten reactions in each compartment with rates given like so. Okay, the forward rate, the backward rate, and this is the catalysis rate. Yeah, so this is the traditional Michaelis-Menten reaction. The Michaelis-Menten reaction tells you that an acceptor, namely the process proteins, let's say, any of these P's, plus a glycosyl donor, there's a sugar, small sugar group, plus the enzyme, which acts on this, gives rise to this following things. Okay? At the end of the day, it will give you a glycosylated acceptor. The acceptor gets modified by adding a donor. You get a nucleotide out, and you get an enzyme. The enzyme comes out again. Okay. Okay. Now, what it turns out that the number of glycosyl donors in each cisterna is chemostatic. Okay, this is an experimental fact. So you hold as constant the number of glycosyl donors. If it's consumed in a reaction, you supply one more. Yeah. Okay. And these are the reactions written in this same thing written like so is the chemical reaction that happens. So given a schematic like this, of course, it's easy to translate this into equations. Yeah? The equations will be how the concentrations of each of these chemical species versus evolves in time, and it's the chemical species in a given cisterna evolves in time. Okay. Okay. So now we have to go to the next part, which is the specificity of the enzyme substrate reaction, and here is slightly more involved. So let me say the following. Remember, we want to allow for the enzyme, there are only 15 enzymes and 10 to the 4 reactions. Okay? So clearly, a given enzyme is catalyzing many substrates. Yeah? Many reactions which involve different substrates. So how do we model that? And we model that by the extremely well-known MWC model, which is schematized here. Imagine I have an enzyme in a native state of size or shape, okay, quantified by this script L alpha J. J is the cisternal index, and alpha is the enzyme index. So a given enzyme type can have a given shape in a given cisterna. And how does that happen? Remember in the first lecture I said every cisterna has a different physical variable. It has a different pH, right? Okay, so the pH is known to affect molecular flexibility, chemical reactions, and so on and so forth. Okay, so in this model, we'll assume that the native state enzyme has a rest length which is given by L alpha J. Okay, and what we'll say is like a distortion model. This enzyme, okay, will react with that substrate, okay, only if it can distort. Okay, so this one has distorted, and now you can see there's a perfect match. Yeah, this distortion is controlled by this parameter sigma. So if sigma here, called the specificity, is small, then it will allow for large distortions because it will allow for large deviations from Lk and L alpha. This guy, right? So this is like an elastic model. This is like the elasticity models. Yeah, you understand, no? It's just Hooke's law I'm using. So this is telling you if sigma alpha is small, it's a weak spring. Okay, so I can deform it more. If sigma alpha is large, then it's a very stiff spring, so I can't deform it. Okay, so sigma here is the specificity. So you can see that sigma characterizes specificity. Sigma is small; it's a promiscuous or non-specific enzyme. Sigma is large, it's a highly specific enzyme. Okay, so this is a nice elastic distortion model of the enzyme, which in spirit is like the MWC model. Okay? What this means is that the enzyme efficiency now, because sigma and L alpha, this quantity L, has index J, which is a cisternal index, it means that the enzyme efficiency depends on the cisterna. Okay? In some cisterna, it may not work very well, may work very poorly. In some other cisterna, it will work well. Okay? Okay. Now, why does the enzyme need to keep the entire target? Yeah, this is a cartoon. Yeah. So this cartoon was actually taken. There's a paper by Swiss on this, but this picture is taken from Swiss. Their model is different from ours. Yeah. There are some details here. For instance, this is a one norm rather than a two norm, and so on, to make the calculation simpler. But this will work for any kind of model that you can make. Okay, so now given the features that I discussed, now it is time to write down what the chemical reaction kinetics is. And this is the chemical reaction kinetics, which follows from this set of reactions, right? This is mass action kinetics. Okay, and this is what you get. Okay, the change in CK. K is the kth species, the reaction which goes from 1 to Ns. J is the cisternal index, which goes from 1 to Nc. Okay? And how does that change in time? And it has this standard Michaelis-Menten form. Looks more complicated than the Michaelis-Menten, but this is the standard Michaelis-Menten form. And these terms, namely V, M, and P, okay, I'm going to rewrite in terms of this huge vector. Okay, M vector will stand for this quantity. Okay, this is the backward rate, the catalysis rate, the forward rate, which depends on all these indices. The index representing the chemical reaction K, J is the index representing the cisterna, alpha is representing the enzyme. Okay, so you have M, V, and L, which are the parameters in this chemical reaction. This chemical reaction is written when you don't have any internal cisternal transport mu. When you have it, of course, you can augment that. Okay, I'm not going to write it because it's a bigger expression, but it's easy to do that. That'll change J to J plus 1 and J minus 1, depending on where it moves. Okay. Now this quantity here is the binding probability, and it's related to the elastic model that I talked about earlier. Okay, so now we have completed, I think, that's it. We have completed the description of the channel. The channel properties are prescribed by partitioning Ne enzymes in Nc compartments, by providing a sequential Michaelis-Menten reaction in each compartment with enzymes of variable specificity, which uses the MWC or elastic model. We then provided the chemical reactions in the last chemical reaction kinetics shown in the last slide. We realize that the reaction efficiency depends on the cisternal index J, and therefore is compartment dependent. And one could think of the physical variable, namely pH, as distinguishing the different compartments. And finally, I provided you with the chemical rates given by V and M, okay, and transport rates, which are mu, okay, which I haven't written down. Because of it's a big, yeah. Why is it that I can't figure out? Yeah. Yeah. So far, you have written only deterministic. Right. Yeah. Correct. Right. In the calculation I'll show you today, there's no stochasticity. You're right. But I could, in principle, solve it by Gillespie. We haven't done an exhaustive calculation on that, but that's what we need to do. Remember, this is chemical noise, so you write down the chemical master equation. You can't just write a Langevin equation with additive noise. Yeah. I mean, for small noises, it will work. You can do that to test the. Because this itself, is like a probability distribution. You're right, technically you're right. I should be also doing that. I should also include this noise. But you see that here I have a distribution also, which I'm going to, the distribution of the various numbers of CK will give you a distribution. No? Yeah. But you're absolutely right. This one does, this one at this, this is our first attempt at solving this, and this doesn't have stochasticity. I mean, you can appreciate that doing a Gillespie on so many reactions is complicated, but we'll do it, yeah. I mean, being complicated, as Pauli said, being complicated is no excuse. So, yeah. This one? Sorry. Yeah. So if I look at just one compartment. Yeah. The equation for the CK. Oh, yeah, yeah. Which one? No, sorry. I'm doing something wrong. Forward. Yeah. This one? Yeah. Yeah. So let's say you have only one compartment. Yeah. So the index K means how many reactions this protein has already had. Yeah. But so I have one compartment and I put all the enzymes there. Right. But within, but the C of K of the Kth reaction could have had many different types of glycans. Right. So it's not. So you now have to have another axis to say how many building blocks were at point 1, how many at point 2, like which ones were at point 1. Yeah, I'm not. So this is a cartoon chemistry. So I'm not going to tell you what are the. So you should think of the C1 and C2 as being providing. It doesn't need to be linear polymerization.
No, that's
yeah.
But you don't say which kinds. You only say
No,
how many.
That's true. So that's a big failing of this, and we are working on a more elaborate chemistry model where we'll have different.
You didn't distinguish between the types of enzymes here.
No, that we do, because the enzyme features are encoded in this. Okay, so yeah.
How probable I am.
Yeah.
Add one more unit. But But at the end of the day,
no, but
lumped all the proteins that have 10 units.
Yeah.
But you didn't care if 5 are red and 5 are blue or blue.
Right.
This information you completely obliterate.
So far, yeah.
Okay.
Correct.
But that could be
Oh yeah, absolutely. It's an important. Yeah, that enhances the complexity. But even the simple model, I'm just saying, can be quantitatively carried out, and we can see.
You could have used only one enzyme.
No, you can't. I'll show you that. It's very sensitive to enzymes. Let me go through and then. Remember, I want to reach the target. That's the most important thing. So the target can be a complex target. So let me just say, okay. So at the end of the day, so this is the steady state concentrations that I obtain by setting this left hand to be 0. Okay. And this is the formula where the intercisternal transfer rate has been restored here. Okay. So this part has to do with the chemistry in each cisterna or compartment, and these mu's have to do with the intercisternal transport rate. Okay. Just to remind you that J here is the compartment or cisternal index. K is the glycan index, the reaction index. Mu is the transport rate, and M and V are enzyme parameters. Okay, because they involve the rates, and they're involved in the Miklas-Menten kinetics. Okay. So now I want you to recall that definition of the ratio of dKl to the entropy. This F, which I call fidelity. Okay. So this is the fidelity, the ratio of the KL divergence between the synthesized distribution C, which I compute from here. Okay, which is where J is the last cisterna, and the target distribution C star. Okay, I follow the definition that I gave earlier, and that's on this side. But since I know I'm given CK star because I know the target, okay, and the target is a complex target, and I've computed what CK is because that is what you get from your reaction kinetics. I can actually make an estimate of this FC. Now the task is that the parameters that I am, that this of course depends on all parameters. It depends on sigma, the enzyme specificity, the number of enzymes, the number of compartments, and the target for a given target. Okay, and it also depends on the rates. Okay, rates and some other properties of the enzymes, transport rates and chemical rates. Okay, so I can take this quantity, which I'm calling synthesis error, and I can minimize it with respect to all these other rates. I can minimize it with respect to the transport rate, with respect to the chemical reaction rates, with respect to the partitioning, and so on and so forth. And I can ask, I want to try to minimize this and achieve an optimal value for this quantity fidelity. Yeah? And I want to see what are the values of these optimized rates. Yeah? How do these optimized features of this channel show up, and how do I analyze this optimized feature in terms of number of enzymes, number of compartments, and specificity. So I can study by this quantity, I can study the trade-offs between the number of cisterna, number of enzymes, and specificity in order to achieve target glycan distributions of a desired complexity and diversity. Okay, so that's clear? That's the exercise. Okay, yeah.
If you have a Markov chain with a limiting distribution, usually the entropy will increase and the KL divergence will decrease, right, over time, not over the parameters, over time, right? If you have a limiting distribution and any arbitrary state is going there, so fidelity seems to have a definition of scale divergence divided by entropy.
Correct.
So overall fidelity would
decrease
over time.
Correct. Not over time. You want to. So we are taking the steady state. So there's no time now.
There's no time.
No time. But what you want to do is that you want to do it. You want to compare the performance of one channel over the other, and you want to see what is the most optimized channel.
I was asking a little different question.
But you could ask in time. Correct.
You were reaching there. Towards the steady state.
Yeah.
Then over time also fidelity has to decrease.
That's.
Now you're asking once you reach that steady state.
Yeah.
Over the parameters, what's even the most minimum?
Right. Yeah, in some sense, there's a separation of time scales here. One is the time scale over which this processing is happening, the channel is operating. That's a faster time scale compared to the evolutionary time scale, which sets the target. Okay? So you could, for instance, ask a question where the environment is changing in time, right? Which then gives you a slowly varying target distribution that you want to achieve. And then you could ask questions of the dynamical response of such a system. That's indeed a very interesting question to ask. Okay, so I just wanted to state this, that this minimization is highly non-convex, okay, and therefore is very, very hard because the concentrations here are given as some, it's not easy to relate the concentrations to these parameters, okay. But what Alkesh did was to prove a very, very clever theorem where he actually applied a nonlinear transformation on these enzyme rates, and that achieved the fact that this minimization of optimization was easier to do. Okay, that was actually a big stumbling block for us for a long time, and this guy Alkesh did this very non-trivial job of this. Okay, so I'll now look at the results. Okay, so now the logic of compartmentalize. I want to now explore these various results as a consequence of our analysis. Okay, we are looking at so what we want to do is to look at the synthesis error, namely F, as a function of the number of enzymes and the number of cisternae. Okay, and what you have here is NC, number of compartments or number of cisternae, versus number of enzymes. Okay, NC, remember, varies from, in this case it goes from 1 to 10. You can, and here NE is 1 to 25. Okay, so they're roughly in the same usual as you see in a cell. Okay, and what the color scheme here is that fidelity F. Yeah, so what you want to achieve is, of course, a low fidelity, right? You want to move towards that, and you can see that low fidelity for a given complex target distribution. This is from the human neutrophil data. Okay, comes here for a very large value. So it sort of comes in this region. Okay, where you have larger number of NC and many enzymes. Okay, So a more complex, whereas a simpler distribution, suppose I've just given it a Gaussian, a single Gaussian, then that can be achieved by having a very small number of compartments, one, and a very few number of enzymes. Okay, so it really depends on the complexity of the data that I want to achieve here. Yeah? Okay, now. So one of the results that we find is that the more complex target requires higher number of enzymes and a higher number of, or a higher number of cisterna. Okay, remember there is a cellular cost, a genetic cost to making more enzymes. Okay, so it's not that the number of enzymes are large; they're of the order of 15. Okay, and so given that it's quite expensive to make more and more enzymes, more and more different kinds of enzymes. Having more compartments, okay, is a way in which you can achieve a complex target. Okay, so that is one. The next is the logic of enzyme promiscuity or enzyme specificity. Okay, and what I plot here is again the normalized KL divergence versus enzyme specificity parameterized by that sigma, okay, which is the spring constant in that elastic model. Okay, remember low sigma is low specificity or promiscuity, high sigma is highly specific. And one draws this graph for different values of the number of compartments. Okay, so what you see is that if you have one compartment, in order to achieve a highly complex target, you require quite highly specific enzymes. Okay. By going to larger number of compartments, you allow it to have lower specificity of enzymes. Okay, so the enzymes can be quite promiscuous. In other words, the enzymes should not be too specific. You see, the minima is neither 1 nor 0. Should not be too specific or too promiscuous when you have a larger NC, okay, for a complex glycan distribution. Moreover, so this is the graph, set of graphs for high complexity. This is the set of graphs for low complexity. For low complexity, you can see that the minima is pushed towards smaller values of sigma. Okay, okay. And it's roughly independent of the number of compartments that you have here. Okay, the minima is roughly independent of the number of compartments that you have. Okay. The optimal enzyme specificity increases on increasing the number of compartments or enzymes. Okay, so that's the other feature. A lot of these can be, in principle, experimentally tested. So, okay. So now, what happens when you repartition? So here's an exercise we did. This is the cisternal index on the x-axis. Once the first cisterna, second cisterna, etc., etc. This one is the reaction index. Is some for each chemical reaction, you define something like a reaction efficiency. Okay, and this is for the set of each of these. This thing is all the chemical reactions. For this chemical reaction, namely the second one or fifth one, is very highly efficient. With low efficiency here, high efficiency in that chemical reaction, namely 85, and so on. And you can do this for different cisterna. Okay, this is the complex target distribution that I display that I want to achieve. Okay, this tells you the abundances for different different glycan index. Okay, upon optimization, as I discussed earlier, upon optimization, this is the glycan profile that I actually synthesize, and you can see it's quite close to. Close to this. This has been optimized to get the KL divergences to be small within a certain error. Okay. Now, yeah. So this is what I'm saying, that you're optimizing over the whole range of partitioning, rates, etc., etc.
The rates are not the same in our system.
No. Because the pH is changing. So
the same enzyme
can
function differently.
Right, in different systems. Yeah.
So it's like having another kind of enzyme.
It's sort of like having another, but this is a property of enzymes. Enzymes. Yeah. Right. Yeah. It's not the same as number of enzymes because the number of enzymes involves the genetic cost.
Right, but they
Yeah.
function completely different.
Completely different because they're in different environment. Yeah. So enzymes are known to do this. Yeah. That's right. That's right. That's right. Okay. Now, if you repartition, so I take this optimized. Right. This is a good thing. Now I'm going to repartition thing. I'm going to exchange 4, okay, and 2. So I'll exchange the enzymes from 4 and 2, right? And that is what the exercise here. I've just exchanged it from 4 and 2. Now you can see that 4 closely resembles 2 and 2 resembles 4. Okay. Now we can run the dynamics, and you find that, so we just run the dynamics here, and we find that the glycan profile, keeping everything else constant, okay, keeping everything else optimized as in that, the glycan profile that I achieve is very, very different from the target. Okay, so the simple act of repartitioning has totally changed the glycan distribution. So the partitioning is crucial. The cell has to have a strict control over the mechanism by which it partitions enzymes into the system. Okay. Yeah. Okay. Finally, let's talk about the geometry of the fidelity landscape. Remember, we want to optimize this this synthesis error, right? But this synthesis error is a highly non-convex optimization, correct? So there will be lots and lots of minima. It's a very rugged landscape, right? It's like glasses or something like that, right? It's a very rugged landscape. So what you have to do now is to have initial conditions, choose initial conditions from very, very, very random thing, and see if you hone in into minima. But since there are multiple minima, there are a degeneracy of solutions. Okay? In fact, there's a very, very large degeneracy of solutions. Yeah? And some of these minima are discretely separated from the one that you achieved. Some are continuously separated, are continuously connected to the one. Yeah? Okay, and now you can ask the following. Suppose I go to the optimum, the very minima that I have, and now I've got, now that optimum will tell me what are the optimal parameters. It'll tell me what is the optimal number of enzymes, number of compartments, specificity, chemical reaction rates, partitioning, right? It'll tell me these are the optimal values. Now I'm going to tweak these optimized values of these parameters ever so slightly, right? If I tweak them, that will clearly change the synthesis error, right? But how much does it change? In some directions, it won't change too much. In some directions, it might change a huge amount, right? So this gives rise to the notion of stiff and sloppy parameters. Stiff parameters are those where if I make a tiny change in the parameter, that will change the landscape entirely, or that will change the value of the synthesis error by a large amount. Sloppy ones are okay, where I vary them slightly, there's hardly any change. Okay, and the way to quantitatively look at this is to look at the Hessian of this surrounding the minima. Okay, the eigenvalues of the Hessian will have some information about the stiff and sloppy directions. So what we have here are the eigenvectors, okay, of the Hessian, and what we find is that they are clubbed into distinct sectors. These set of you can see these set of eigenvectors correspond to have a large projection along the cisternal transport direction. Okay, these set of eigenvectors have a large projection with the chemical reaction rates. Okay, R is the chemical reaction rates. L here is the shape of the enzyme, the shape variables that dictate the enzyme. And finally, sigma, which is the last eigenvector, 37, okay, has a very large eigenvalue. Yeah? And that corresponds to specificity. So the stiff directions are ones where the eigenvalue of the Hessian is very large. Okay? So you can see that the specificity for this n equal number of enzyme, number of compartment equal to 4, the specificity is a very stiff variable. Yeah? Make slight changes in specificity that totally alters the synthesis error. Okay. Okay. Here are the eigen. Again, replotted the eigen in this mu. So I'm now collecting it in these different sectors. This is the transport. This is the chemical reaction, enzyme shape, specificity. You can see that this one. Remember, this is a log scale. This one has is the largest eigenvalue, and that corresponds to the specificity. Now, okay, now should I talk about this? Yeah, I don't, yeah, maybe I don't need to talk about this. Okay, so now, the bottom line of the following, that the eigenvalues of the Hessian of the error, of the synthesis error, at the optimal point shows sloppy directions in the directions corresponding to the reaction rates. Okay, it's very sloppy in the reaction rates. That's the lowest. It's very stiff in this what is this called, the specificity direction. Yeah? Okay. What was this? This was, yeah, this was plotting the mean of these quantities. Okay, in each sector versus the number of compartments. Okay, and what you can see is that in the specificity compartment, you're very high. Okay, the eigenvector is very stiff in the lower cisterna, but you're allowed to be slightly less stiff as you go up. The shapes have to be very high in the higher number, when you have more number of compartments. Sorry, when you have more number of compartments, it's high. And here the specificity, the mean, I made a mistake. The mean eigenvalue in the specificity sector is sort of almost independent of the number of compartments. Okay? Yeah. Yeah, I'm done. So because of this, that there are few number of stiff directions and a lot of sloppy directions. Remember, there are 10 to the 4 reactions. So all those parameters associated with that are sloppy. The landscape, the fidelity landscape looks something like this, like a valley. Okay, where the sloppy directions, namely the chemical rates, are along that ridge there, along that valley, and the stiff directions are along this direction. And the stiff directions correspond to specificity, transport rates, and partitioning. And because they are stiff, these must be under very strong cellular control. Yeah? And it'll be interesting to figure out what are the kind of cellular control mechanisms that control the values of these optimal parameters. Okay, so to summarize, I'd like to reiterate that the glycan profile in real cells are highly complex. We had a definition of complexity in terms of the Gaussian mixture model. We studied the trade-offs between compartment number, enzyme number, and enzyme specificity that achieves this complex target distribution. Complex glycan distributions require multiple compartments, precise enzyme partitioning, and promiscuous enzymes. The latter allows for evolvability. What this analysis suggests is that the control of cisternal number, compartment number, must involve a coupling of non-equilibrium assembly of cisterna, as I described in the last 2 lectures, together with enzymatic reaction kinetics. Okay? And so this is what we would like to do in the future, because our study here suggests that you need to couple both the enzyme kinetics and the kind of membrane morphologies and kinetic theory that I talked about earlier. So before ending the talk, I just wanted to sort of put this thought in your mind, that a way to look at how to encode molecular information is by the following flow chart. In this particular case, you have a cell with a given identity, let's say skin cells, etc. Let me call that space Z. This is a space of cell type identities. Each cell type gives rise to a set of molecular units, which are these processed proteins that are synthesized in the ER. This then goes through a synthesis channel, the glycan synthesis channel, which is the Golgi. And finally, what you get is a signal that is displayed at the source, namely the plasma membrane. Okay? So this is the kind of information channel that is being that gives rise to an encoding, okay, a molecular encoding of molecular. The kind of analysis that I talked about, as you saw, was a high-dimensional optimization, many parameters. Okay, and we used a combination of information processing and systems biology. But very interestingly, the number of stiff parameters is small. That is, the stiff parameters is a low-dimensional space. Okay? So this is the paper in which this is described. In the future tasks are to look at the kind of question that you asked, namely, how do you study a dynamical glycan profile in a time-varying, time-varying niche? And there are both fast and slow responses, and this will be an interesting study. We would, in the calculation I showed today, there is only forward transport of material. What is the role of retrograde transport? Is there some error correction that can be thought about? One could ask this question, which is sort of related to the kind of question that Amitabh had. Namely, how far from equilibrium do you need to drive the system to achieve a complex code with a high fidelity? This will involve energy, accuracy, cellular cost trade-offs. And finally, it will be interesting to see what is the role of intracellular trafficking in non-equilibrium proofreading to reduce coding errors in a time-varying environment. Okay? And so with that, I just flash these two references that you might like to read. And just to acknowledge the people who have done the work. Alkesh Yadav, a student of mine who's now graduating, has done all the work here in this, together with very strong collaboration with Garud Iyengar, who is an information theorist, an applied mathematician at Columbia University. And this work is also done with Pierre and with Conta. Okay? And thanks a lot for the attention.
I think we won't take any questions now during lunch. So now I would welcome Professor Debashish Chowdhury, who would close this meeting on behalf of our.
Okay, so I have the pleasant job of saying a few words of our appreciation before we close this ICTS program formally. So first of all, I'd like to thank all the invited speakers for giving excellent lectures, and then also giving the pedagogical introductions for which we had requested them. So a big round of applause for them. We would also like to thank all the participants for their active participation, and there are several thought-provoking questions which I am sure will initiate new projects. So thank you for those questions and your active participation. I would like to thank ICTS Center Director, Professor Rajesh Gopakumar, and the program's Dean of ICTS, Professor Abhishek Dhar, for supporting this. And whenever we needed their advice or some help, they immediately responded to those. So on behalf of the organizers, we thank them for making it possible. Now, of course, the program office handled all this administrative work. So program office of ICTS, particularly the administrative head of the ICTS program, Ms. Aruna Mahendarkar, and particularly, very specially, Ms. Kavyashri, who must have written to all of you. She handled almost single-handedly the entire administrative part, so we thank them. Finally, somebody who was not visible here, but whoever organizes ICTS programs should thank him, and this is the founding director of ICTS TIFR, Professor Spenta Wadia, for having the vision of starting such an institute and initiating such ICTS programs. So we should all thank him for this. So thank you all, and that's all from the side of the organizers. So now we'll close this ICTS program, but discussions will continue, and we'll now go for lunch. Thank you.